Today we shall continue our discussion on this lecture series on archegoniates. We have come towards the fag end of this entire series, and in today's lecture we are going to understand one of the most important and abundantly spread group of gymnosperms, that is coniferales or conifers. We shall try to understand this group with the help of a type genus also known as Pinus. We shall see the morphology, anatomy and reproduction in Pinus, and with the help of these aspects we will try to understand and get some idea about the general feature of this entire group called as coniferales or conifers. So, as far as the group Coniferopsida is concerned, it consists of 5 orders: Gingoales, Zecanovasciales, Corditales, Volziales and Coniferales. Among these 5 orders, Coniferales constitute more than 3/4 of all the presently growing gymnosperms, and they form a significant part of the world flora today. This order alone comprises of 52 genera and more than 550 species, which are grouped into 7 families, and these families are Orochariaceae, Cephalotaxaceae, Cupressaceae, Pinaceae, Podocarpaceae, Taxodiaceae and Taxaceae. As far as coniferales are concerned, some genera are widely spread all over the globe, while considerable number of taxa are endemic and show restricted distribution. This implies that it is an ancient group which was formerly widely scattered somewhere around mid-Mesozoic era, and voluminous forests occurred in Europe, but their successive decline led to their isolated distribution. Let us talk about some of the most common features of conifers. So, most of the conifers, they show xerophytic characteristics. They are woody perennials which have long and dwarf shoots. The leaves in conifers are dimorphic, that is, foliage and scale leaves are found. The tap roots are associated with mycorrhizal fungi. The wood is pycnoxylic, that means it has very less or no parenchymatous tissues, and secondary xylem lack fibers. Male and female cones present on separate branches but on the same plant, hence they are monoecious. The pollen grains may be winged or non-winged, and they produce non-motile sperms. Now, as far as the family Pinaceae is concerned, Pinaceae constitutes the most widely represented family of the conifers. It is also the largest extant family of conifer families. It has 11 genera and 232 species. These 11 genera are Abies, Cathaya, Cedrus, Cataleria, Larix, Nothosuga, Picea, Pinus, Pseudolarix, Pseudotsuga and Tsuga. The family Pinaceae composes of highly resinous shrubs or trees. Leaves are evergreen or deciduous, usually with resin canals and needle-like or oblong and spirally arranged group of spurs on the shoot. Male and female cones occur on the same tree. 6 genera are found in Indian subcontinent, which are Pinus, Tsuga, Picea, Abies, Cedrus and Larix. As far as the male cones are concerned in the family Pinaceae, they have an axis with numerous spirally arranged microphyls. The female cones have bracts scales and ovuliferous scales. The distinct feature of the family can be summarized as follows. Most of the parts are spirally arranged. Microphylla bearing pollen sacs are arranged abaxially. Pollens are mostly two-winged. Ovuliferous scales are free from the bracts scales for most of their length. Two ovules per scales are present on the adaxial side of the scale, and seeds are mostly winged. Now let us talk about the genus Pinus, which is the most well-known representative of this family. It is a largest genus within conifers with about 115 living species. Pinus is a beautiful large tree with widespread branches arranged in whorls, giving the tree a pyramidal appearance. On maturation, the tree starts losing their symmetry and crown becomes round, flat or spreading. The pines form voluminous pure forests or even form mixed forest in association with broadleaf trees such as quercus and other species. Let us talk about the characteristic feature of this genus Pinus. Pinus is monoecious. That means male and female cones are present on a single plant. The leaves in Pinus are needle-like and produced in a cluster of 2 to 5. The leaves and other parts of the tree possess resin canals that exude resin. Some of the plants are known to produce turpentine and rosines also. The taproots are associated with ectomycorrhizal fungi. The wood is compact and of pycnoxylic type. Here we can see a typical Pinus tree. It's a long woody tree with a conical shape, but as the tree grows old, the upper pointed part gradually rounds off. On a single tree, the male and female reproductive structures, also known as male cones and female cones, are found, and the leaves are of two types, that is the scaly leaves at the base and the foliage leaves. And these leaves, they are arranged in clusters. These clusters are borne on spur shoot, and these clusters, the number of needles per cluster can vary from 2 to 5. Pines are widely distributed in northern Europe, North and Central America, subtropics of North America, the Canary Islands, Afghanistan, India, Myanmar, Pakistan and Philippines. And they spread up to Indonesia after crossing the equator. In tropical areas, they occur at subtropical or high temperature altitudes also. In India, Pinus is confined to hilly regions, usually at a height of 1500 to 2500 meters. 7 species of Pinus occur in India, of which 4 are restricted to Himalayas. The Pinus tree is a sporophyte and is differentiated into root, stem and leaves. As far as the roots are concerned, Pinus exhibits 2 types of root system. The long roots having potential for indefinite growth constitute the main root system, while the branches with restricted growth and relatively short life are the short or dwarf roots. Here you can see a typical root and their branching pattern of a Pinus tree. They grow laterally and they gradually tapers down towards the center of the earth. In some of the species, they are much dense and fibrous, and in many of the species, they also grow in a lateral side so that they could provide a proper anchorage to that entire length of the tree. The mycorrhizal fungi occur in association with most of the conifers. In Pinus, it is well developed and exhibits well developed ectotrophic mycorrhizal association with over 50 different species of fungi, generally belonging to families Bolitaceae and Agaricaceae. And these Bolitaceae and Agaricaceae belong to Basidiomycetes. The dwarf shoot roots, they divide dichotomously and become modified into mycorrhizal system after fungal infection, when the entire rootlet is covered by mycelium. The fungal hyphae penetrate the intercellular spaces in the cortical cells of the root, forming the Hartig's net. It is seen and understood with the help of these diagrams. Here we can see that on the dwarf root, the fungal hyphae create an entire sheath or entire covering, also called as mantle. These associations are ectotrophic in a sense that they never penetrate any of these cells, but they spread their mycelium in the intercellular spaces. And this cortical cell, they are completely covered by the spread of mycelia. These mycelia, these fungal hyphae, they are connected with each other, and this spread goes up to endodermis, which has Casparian strip, and this spread does not go beyond endodermis. This interconnected fungal hyphae, they form a net-like structure, also termed as Hartig's net. Here we can see with the help of a diagram that this is the central cylinder of vasculature. This is the endodermis with Casparian strip, and in the outer cortical cells, the fungal hyphae, they are found in between the cells, and these interconnected hyphae, they form the Hartig net. An outer hyphal sheath, that is also termed as mantle. So the pine fungal relationship is symbiotic in nature. The pine tree benefits by the increased absorbing area of mycorrhizal roots and enhanced absorption of soil nutrients, while fungi gain most of their sugars from the living roots. In a few pines, seedlings that are grown in sterilized soil do not grow well until the fungi are introduced and allowed to develop in soil. It is also have been found that these fungal associations, they help in the absorption of phosphorus from the soil, and thus the pine tree gets all the different kinds of nutrients from the soil with the help of these fungal hyphae. The stem happens to be erect. In case of pinus, it is cylindrical, mostly woody and covered with irregular scaly bark which peels off later on. There are two types of branches or shoots: the long shoot or the branches of unlimited growth. It arises on the main stem as lateral buds in the axils of a scale leaf and terminate in apical buds. The apical bud remain enclosed in bud scales. These lateral buds grow horizontally on the main stem to a certain length, and this is referred to as nodal growth. The spur shoots, when they fall off, leave a scar on the stem, whereas the dwarf shoots or branches of limited growth, as they are also called as short shoots or brachyblasts or foliar spurs, they are borne on long shoots and arise in axils of scale leaves. Each dwarf shoot bears two opposite scaly leaves called prophylls, which is followed by 5 to 13 spirally arranged scaly cataphyls. The long shoots bear the scale leaves or cataphyls, while the dwarf shoots bear the green acicular foliage leaves, which are also called needles. Each species of pinus has a fixed number of needles. Later on, the scale leaves and the spur fall off, leaving a scar which looks like a crescent or half moon with a dot in the center. You can see in the diagram the two types of leaves, the scaly leaves and the foliar leaves. The foliar leaves, they are found on the dwarf shoot, whereas the scaly leaves, they cover the long shoot in case of pinus. The leaf of pinus displays xerophytic characters such as thick cuticle, sunken stomata, sclerenchymatous hypodermis, simple vasculature and distinct transfusion tissue. The pine leaves on dwarf shoots are of two types, that is scale leaves and foliage leaves. The scale leaves are of protective nature while foliage leaves are needle-like. The needle number is constant for a species and is used as a taxonomic character. For example, Pinus monophylla has one needle, Pinus sylvestris, Pinus nigra, Pinus marcusi have two needles, whereas Pinus roxburghi, Cassia gerardiana has three, and Pinus wallichiana and excelsa has 5 needles per spur. We can understand the number of needles per spur, which are different in different species. Here, in case of Pinus monophylla, one spur gives rise to a single needle, whereas in case of Pinus sylvestris, a single spur has 2 needles. During the early development, if we cut a cross section, we will see that these are nothing but the modified single spur which have been divided into 2 equal halves. Similarly, in a spur with 3 needles, each needle has a triangular shape, whereas in case of Pinus wallichiana, where a single spur has 5 needles, the entire symmetry is as such that each of these needles they look like a piece of a single circle. The primary leaves, which are always single, appear soon after germination and function as foliage leaves. The secondary or the permanent leaves or the needles are produced on short shoot or brachyblasts in clusters in the axis of scale leaves. Each cluster, called fascicle, forms a cylinder or collar if the leaves are clutched together. Here in this diagram you can see the scale leaves and foliage leaves. The foliage leaves, they come out of a single spur, and these spurs, they arise from the axials of these scale leaves. Now let us talk about the anatomy of stem. As far as the anatomy of Pinus stem is concerned, the stem bears resemblance to that of a dicot stem. In a transverse section of a young stem, ridges and furrows are formed due to at-pressing of surrounding leaves. So it appears wavy in outline. The epidermis is single layered and very much cuticularized. Below the epidermis, multi-layered hypodermis comprising of sclerenchymatous cells are found. The hypodermis is followed by the broad parenchymatous cortex is followed by a ring of discrete, conjoint, collateral and open vascular bundles. The number of these vascular bundles can vary from 6 to 8 in number, and they are surrounded by common endodermis, pericycle and pith. Bundles are separate from each other by broad medullary rays. The xylem of the bundle is endark, that means they have protoxylem towards the center and metaxylem towards the periphery. The first formed protoxylem elements show annular or spiral thickenings, while the metaxylem mostly displays reticulate or even pitted thickenings. Raceme ducts with an inner lining of thin-walled epithelial cells are present. After a wound is imposed, many raceme ducts are formed around it. Let us try to understand the cross-section of a young stem of Pinus with the help of a diagram. So, the outermost layer is termed as epidermis, which is mostly ruptured because of the uneven thickening. Below the epidermis is found the cortex, which has raceme canals. The cortex is made up of parenchymatous tissues. Below the cortex or towards the center from the cortex is found the endodermis and pericycle. Inside the endodermis and pericycle are found the vascular bundles where the phloem is towards outside and xylem is towards inside. Each bundle are open, that means they have cambium tissues in between xylem and phloem. In between two vascular bundle are found medullary rays. Now let us talk about very briefly about the secondary growth of stem in Pinus. Before the secondary growth initiates, the fascicular cambium and interfascicular cambium join to form a complete ring, and thus the cambium cuts off two types of cells, the fusiform cells and ray initials. We can discuss this. The fascicular cambium, which are found between phloem and xylem, and then there is interfascicular cambium, which are formed between the two vascular bundles. So there is an entire ring of cambium which is formed, and this ring now separates the stele into two halves. Now this cambium gives rise to two types of cells, the fusiform cells and the ray initials. The fusiform initials are elongated with tapering ends and are tangentially flattened. They give rise to the axial system, that is xylem and phloem. The ray initial, on the other hand, are small and isodiametric and form the radial system, which is composed of rays. The cambium produces a continuous cylinder of secondary xylem towards inside and secondary phloem towards outside. The cambium also cuts off parenchyma cells, which produces secondary medullary rays, which are unicerirate and thick walled. We can understand this secondary growth with the help of this diagram, where we can see that the cambium, it cuts off secondary phloem towards outside and secondary xylem towards inside. This growth rate changes with season, and with every passing year we can see the formation of summer wood and formation of winter wood. So the summer wood is early wood and the winter wood is late wood, and we can find multiple growth rings being formed here. And the phloem, when it grows, the secondary phloem, that it later on gives rise to some part of the cork or periderm. So by now we have understood the basic structure of various organs of Pinus. We have gone through the distribution of Pinus, the morphology, the root structure, stem and leaf of Pinus, and also we have seen in a much greater detail the anatomy of root, stem. We shall continue our discussion over the anatomy in our next lecture. I hope I have been able to give these concepts in a much greater detail with more elaborate diagrams. Thank you so much.